Well, good afternoon to everyone and good evening for those that are hearing us from Europe. Welcome to the session of this afternoon that is devoted to understanding and modifying genomes. The first speaker of today will be Dr. Anke Becker. Anke studied the PhD at the University of Bielefeld under the direction of our close friend and colleague, Dr. Alfred Puller. Even during her studies, it was quite clear that Anke was a very dedicated scientist with a specific talent for experimental work and also the ability to introduce to the novel technologies, the omics technologies that were rising at that time. After her PhD, Anke stayed at Bielefeld University at the Center for Biotechnology and devoted a lot of effort to build the genomic capabilities in that place. She did also a brief stay at the MIT, and then she became part of the staff of the University of Bielefeld. 3 years ago, 3 years later, she moved to the University of Freiburg, and then she moved to Marburg in 2011, if I remember well, when she became, over the years, the head of the Center for Synthetic Microbiology. Sorry. Anke is a very well-known scientist. She has over 160 papers published, more than 14,000 citations to those papers, with a Dash number of 66. Among the many honors that she has received, she is a member of the Permanent Commission of the German Senate devoted to genetic research, and now she forms part of the review board of the Deutsche Forschungsgemeinschaft. I hope that I say that well. Okay. Today's talk will be about multi-talent rhizobium, versatile, adaptable, symbiotic nitrogen fixer, SynBio chassis, and digital storage medium. Anke, it's a pleasure to have you here today, and I hope that we have you here later, soon, I hope, in person.
Yeah, thank you very much, David, for the kind introduction. I really would have enjoyed more being on that stage and not sitting at home at my desk. I'm starting to share my screen. I hope that's going to work. So I hope you can see my screen and hear me well.
Yes, go ahead. We can see you.
Yeah, I just get this out of the way. So thanks again for the really kind introduction. It's really a great honor to have the opportunity to present research from our lab at the symposium of the 40th anniversary of the CCG. I've started my research career with work on rhizobia in the lab of Alf Puller, as you have mentioned, in the early 90s. So your center was already a major player in nitrogen fixation research when I entered the field. Over my 3 decades of, I think my computer is frozen. Okay, seems to work now. So over my 3 decades of research, I've never lost the fascination with rhizobia, but my research approaches and goals have largely changed over the time. This was also inspired by my scientific environment at the Center for Synthetic Microbiology. SynMIKRO is a joint research center of the University of Marburg and the Max Planck Institute for Terrestrial Microbiology, much younger than your center. It was founded in 29, and I've joined the center about 2 years later. More than 30 research groups with focus on microbiology and synthetic biology are a member of SynMIKRO. And last summer, the center finally moved into a brand new research building. So this is a great research environment for my lab. I've always been fascinated by the ability of rhizobia to adapt to a wide variety of environmental conditions and to change their lifestyles. In the soil, they are exposed to various changing stress conditions, must be able to survive short periods to many years of unfavorable conditions, but also to take advantage of opportunities for growth. The rhizobial cells can either occur in a planktonic state or attach to abiotic or biotic surfaces where they can form a biofilm. And they are capable of inducing and invading nodules on the roots of their host plants and differentiating to nitrogen-fixing bacteroids. In case of indeterminate nodules, this differentiation is a point of no return, but vegetative cells from the infection zones of the nodules can resume growth when released into the soil. This high capacity of adaptation of these bacteria is reflected in their large genomes, high genome plasticity, and large pangenomes. Multipartite genome organization is one of the factors that contribute to genome plasticity and large pangenomes. A high percentage of regulatory genes is another feature that contributes to the adaptation capacity. Today, I would like to share very recent work from my lab on molecular mechanisms underlying the alpha-rhizobial adaptation capacity. This is recent research on how a quorum sensing system integrates each cell's physiological state and need for behavioral adaptation in a collective decision-making process. And I will dive into alpha-rhizobial cell biology and present some of our recent findings on mechanisms that drive establishment and inheritance of systemic cellular asymmetry. Insights from this fundamental research feed into applications that use inherent properties of alpha-rhizobial genome architecture for synthetic biology approaches to engineering of these bacteria. These promote usage of members of the rhizobialis for bioproduct production in biotechnology. We support some projects in this field with commercial partners. Today, however, I will present a test case of using an alpha proteobacterial rhizobium for DNA-based data storage. The first story I would like to present is about autoinducer mediated regulation in Zynerizobium melloti. As melloti has a canonical gram-negative quorum sensing system, the basic layout and regulatory targets of its quorum sensing system has been worked out by the lab of Juan Gonzalez in Dallas and my lab by Matthew McIntosh and Pornsri Charenpanik. In the SYN system, the LOX-I type synthase SYN-I produces long-chain acyl homoserine lactones that are sensed by the LOX-R type regulator XPR. The SYN system has an additional player. This is the transcription regulator SYN-R. Transcription of SYN-I strictly depends on SYN-R and is enhanced by binding of the XPR-AHL complex to the SYN-I promoter. This gives rise to positive feedback that is further promoted by positive regulation of XPR. At very high AHL concentrations, XPR-AHL represses SYN-R transcription and thereby limits the positive feedback. HL-activated XPR has many targets. Most prominently, it promotes isopolysaccharide production and inhibits motility. This regulatory pattern is one of the mechanisms promoting biofilm formation and attachment to roots. Autoinducer production has been shown to be modulated by diverse environmental and indigenous factors. For alpha-rhizobia, this is phosphate limitation, the second messenger cyclic di-GMP and balamone PPGPP, and RNA-based regulation. Effects were also shown for plant root exudates, octopine, and HL degrading enzymes. Some years ago, Lisa Kroll from my lab found another factor that promotes the quorum sensing response of rhizobia that produce long-chain AHLs. This is a FAT-L homologue that has lost the ability to import long-chain fatty acids over the outer membrane but gained the ability to facilitate uptake of long-chain AHLs. Jan-Philipp Schlüter, a former postdoc from my lab, showed that activity of the SynI promoter is heterogeneous in asmelloti populations. He used a triple labeling, the 3 different fluorescent markers to follow in a time-lapse study different promoter activities, and also that of the promoter of a synthase gene. This observation was followed up and confirmed by Vera Bettenwort in her PhD work. Vera further showed that heterogeneity in SynI promoter activity can be also observed in a single bacterial cell. SynI promoter activity in cells carrying 2 identical copies of a SynI promoter fused to 2 different fluorophore genes indicated stochastic independent activities of both reporters. For the wild type, Vera observed a considerable fraction of non-fluorescing cells. In some cells, variation in fluorescence affected both reporters to similar extent, whereas other cells displayed highly diverging intensities from the 2 reporters. In cells lacking the XPR regulator, activation of the 2 promoter fluorophore gene fusions was almost entirely uncorrelated, with most of the fluorescing cells showing fluorescence from either one or the other reporter. This motivated Vera to initiate a study to investigate the molecular basis of this heterogeneity. She teamed up with Simon von Vliet from Basel and the lab of Ulrike Endesfelder. The results and figures that I will show from the study is work of this team with some support from co-workers from the different labs. Monitoring the SynI promoter activity in XPR mutant cells in a time-lapse study showed that cells were usually dark and that fluorescence appeared in individual cells only temporarily. Here are 2 examples, cell 5 and 6, where the signal is increasing and then decreasing over time. However, fluorescent reporters are stable proteins. To more accurately examine changes in SynI expression over time, the expression rate was calculated, and this computational approach revealed unsynchronized sporadic pulses of SynI expression. As intrinsic stochasticity was found both in the wild type and in the XPR mutant strain, next, the central transcription activator SynR was investigated as the most likely source for this stochasticity at the SynI promoter. In contrast to the stochastic activity of the SynI promoter, SynR expression was homogeneous. However, in vivo protein stability assays yielded a half-life of only about 3 minutes for a flag-tagged SynR when produced from its chromosomal SynR promoter. And single molecule microscopy of an M-scarlet SynR fusion protein produced from its chromosomal locus indicated that only about 10% of cells in a population at a given time have M-scarlet SynR spots. To test whether SynR scarcity is a determinant in SynI expression pulsing, 2 strains with slightly reduced and slightly increased SynR expression levels were generated. Single molecule microscopy confirmed that the fraction of cells displaying M-scarlet SynR spots in the 2 strains was altered as intended, reduced and increased compared to the native situation. And time-lapse microscopy indeed yielded about 7-fold reduced and 3-fold increased SynI expression pulse frequencies in these modulated strains. Next, we asked whether pulse modulation also occurs physiologically because pulse modulation might well represent a mechanism for integrating physiological information on the dynamic scale. Phosphate starvation indeed increased SynI expression pulse frequency and the fraction of cells with scarlet SynR spots. A strain incapable of producing detectable levels of cyclic diGMP likewise showed increased SynI pulse frequency, while a strain producing elevated levels of cyclic diGMP showed the reverse phenotype. And the XPR AHL mediated positive feedback raised pulse frequency even tenfold. SynR alone has likely a very low binding affinity to the SynI promoter, but XPR activated by AHL seems to achieve its positive feedback by facilitating binding of SynR to this promoter. In an EMSA, SynR alone did not shift the DNA. XPR did, and XPR together with SynR caused a supershift, of course, all in presence of AHLs. And thus, physiological factors most likely modulate pulse frequency by changing SynR abundance and DNA binding activity. All data gathered in the XPR mutant background indicate a linear correlation between the fraction of cells displaying scarlet SynR spots and frequency of SynR expression pulses. This is true for microscopy and flow cytometry data. In contrast, pulse amplitude in microscopy and fluorescence in flow cytometry do not appear to correlate with SynR abundance. Hence, regulation of SynI expression seem to primarily happen through frequency modulation, and amplitude modulation plays a minor role at best. Differences in SynI expression pulse frequency in single cells should impact behavior on the level of the group, as we will affect the overall AHL production rate of the population. To test this, long-term observations of quorum-sensing target gene expression of wild-type colonies that became already three-dimensional were made. As expected, response onset in strains displaying higher SynR expression pulses could be observed several hours earlier and at smaller colony sizes than in colonies with low pulse frequencies. Further, an AHL indicator strain was used to monitor AHL concentrations in samples of the supernatant at various optical densities. Also in this assay, response curves stacked according to the pulse frequencies of the donor strains. This brings me to our model which we propose. In this model, Asmelotti cells, when adapting to changes in environment or lifestyle, adjust AHL synthase gene expression pulse frequency, which then results in response onset at larger or smaller cell number. I will now switch gears and present some of our findings on mechanisms that drive establishment and inheritance of systemic cellular asymmetry in alpha-proteobacterial rhizobial symbionts. Cellular asymmetries may generate phenotypic variation required for successful exploitation of the environment. Looking at the alpha-proteobacterial rhizobia, a number of cellular asymmetries are obvious, starting with a cell cycle where cell division generates one cell which immediately enters the S phase. This is a cell with the oldest cell pole, which we call mother cell. The other cell called daughter cell, which is slightly smaller, goes through a gap phase before entering the S phase. A further feature of cellular asymmetry is a unipolar polysaccharide, which is produced by many alpha rhizobia. and it localizes to the old cell pole. This is here stained with fluorescent lectin. Unipolar polysaccharide is an adhesin which mediates attachment of cells with their old cell pole to abiotic and biotic surfaces, as shown here for Acrobacterium tumefaciens attached to a root. Another feature of cell polarity is localization of the origin of replication of a chromosome at the old cell pole, and as shown for Zynerizobium meloti, localization of the origins of chromid and megaplasmid in the subpolar region of the old cell pole. Here, all three origins of replication of the tripeptide genomic DNA are labeled with different fluorescent proteins. Then, as meloti cells are growing and dividing under conditions that promote PHB accumulation, this first occurs at the old cell pole. Here, visible by fluorescent labeling of the PHB. This has the consequence that after cell division, the cell with the older cell pole keeps the accumulated PHB. Another prominent feature of cell polarity is unipolar growth of alpha rhizobia, as shown here for S. meloti pulse labeled fluorescent dialanine, which stains the peptidoglycan growth zone at the new cell pole and at the septum. During the cell elongation phase, the peptidoglycan growth zone is at the new cell pole, and with the beginning of septum formation at the cell division site. After cell division, these newborn cell poles become the growth poles. Unipolar growth is likely also the cause of branching in the free-living and symbiotic states in cases of a disturbed cell cycle. This unipolar growth mode, which is rather rare in rod-shaped bacteria, motivated us to ask how localization of the peptidoglycan growth zone is determined in Zynerizobium meloti. What I will present today is mostly work from Lisa Kroll, a great postdoc in my lab, and partly work by Simon Chairperson, who initiated our research in polar growth as a PhD student. The story started with Simon discovering two proteins required for cell growth and showing a characteristic localization pattern to the new cell pole and the septal site. This was the starting point for Lisa, who identified a group of proteins required for cell propagation, all showing one of these two characteristics, spatial dynamics over the cell cycle, and co-localization with the peptidoglycan growth zone. We named these proteins RGS for rhizobial growth and septation proteins. The two patterns Lisa observed are here exemplified by RGSP labeled with EGFP and RGSE labeled with mCherry. RGSP localizes to the growing cell pole during the cell elongation phase. The signal is then building up at the septal site and disappears from the new cell pole. After cell division, the signal is maintained at the newborn cell poles. The other localization pattern here is here of RGSE. It also includes localization to the growing cell pole, but no clear localization to the septal site before cell division. Accumulation to the newborn cell poles only occurs after cell division has been completed. Components of the tall pulp complex also showed this first localization pattern. In E. coli, the tall pulp complex is known to have a role in coordinating outer membrane invagination with peptidoglycan remodeling at the division site. All 11 RGS proteins identified are predicted to be proteins in the inner membrane or in the periplasm, and they are connected by a protein-protein interaction network, which Lisa derived from co-immunoprecipitation and bacterial to hybrid experiments. Computational analysis of the phylogenetic distribution of RGS proteins then suggested that these proteins are conserved in the rhizobialis and mostly absent from other alpha proteobacterial orders. Most of the rod-shaped bacteria elongate by incorporating new peptidoglycan in a dispersed manner along the side wall of a cell, with the filaments of the actin homolog mRAB providing a scaffold for the longer zone peptidoglycan biosynthesis machinery. Interestingly, presence of RGS protein homologs in most cases inversely correlated with the failure to detect mRAB, an mRAB homolog. We therefore believe that presence of RGS proteins is probably indicative of unipolar growth in the rhizobiales. Then the RGSS protein attracted Lisa's special attention because it showed some similarities to the E. coli FTSN protein. RGSS is a transmembrane protein with a conserved spore domain at the C-terminus. Features like transmembrane helix, periplasmic domain, and spore domain are also characteristic of FTSN from E. coli and FTSN-like proteins from alpha, beta, and delta proteobacteria. In E. coli, at the cell division site, FTSN binds to peptidoglycan through its spore domain and in the cytoplasm to FTSA, which is an FTSZ-associated protein. The peptidoglycan polymerase activity of PBP1B is antagonistically regulated by the FTS-BLQ subcomplex and by FTSN. Peptidoglycan amidases play an important role in septum splitting during cell division, and the activity also are important for recruiting the spore domain of FTSN to the septal peptidoglycan in E. coli. Peptidoglycan amidases generate glycan chain 3 of peptide stems. This is called denuded peptidoglycan, and this serves as a binding substrate for the FTSN spore domain. Lisa found that S-Melotti RGSS spore domain and cherry fusion produced in E. coli localized to the septum in the E. coli wild type, but not in an amidase deficient E. coli strain. Lisa then observed that S-Melotti mutants lacking amidase MEC or amidase activator AMCA or producing only a truncated RGS lacking the spore domain showed a low percentage of cells with RGSS foci at both poles in a population analyzed in a snapshot microscopy. This was in contradiction with the localization dynamics of RGSS in the wild type, which only showed RGS accumulation either at one cell pole or at the septum, depending on the cell cycle stage. Carefully tracking the RGSS foci in wild type and mutant cells revealed that in about 30 to 40% of the mutant cells, RGSS mislocalized to the old cell pole, here shown in red and in black, for the daughter and the mother cells, with a higher percentage of mislocalizations occurring in the daughter cells. In this time course here, you see clear cases of such mislocalizations. For example, here and here is the RGS focus at the old cell pole and not at the newborn cell pole. Peptidoglycan growth zone labeling strictly co-localized with the RGSS signal at one cell pole in the wild type and in the amidase mutant. However, PAL, a component of the tall-PAL complex that is a marker for the new cell pole in wild type and in the amidase mutant, localized to the opposite pole in a proportion of amidase mutant cells. They therefore concluded that RGSS mislocalization to the old cell pole correlates with mislocalization of the peptidoglycan growth zone to the same cell pole. This is summarized here. This shows the normal pattern of co-localization of RGSS and other RGS proteins and the peptidoglycan growth zone at the new cell pole during the cell elongation and at the septum during cell division in the wild type. In amidase deficient mutants or in mutants lacking the RGSS spore domain, a considerable percentage of cells showed this mislocalization to the old cell pole, either in daughter or in mother cells, with a preference for mislocalization arising in the daughter cell. To study the mobility of RGSS molecules in the cells, we teamed up with Peter Graumann, who is expert in single molecule tracking. Lisa, a PhD student in his group, observed that compared to the wild type in mutant cells, the mobile fraction of RGSS was increased, and the residence time at the cell pole decreased. This suggests that the amidase and spore RGSS stabilize the positioning of RGS molecules at the growing cell pole. To bring all this data together, we propose a role of the interaction of RGSS with denuded peptidoglycan in faithful positioning of a polar cell elongation complex at the new cell pole. During cell elongation, RGSS is situated at the growing cell pole, and we suggest that its position is stabilized by interaction with MEC-processed peptidoglycan. During divisome assembly, RGSS is recruited to the mid-cell, possibly by one or more divisome components. During cell division, MEC-processed peptidoglycan is formed and probably provides binding sites for RGSS. This is assumed to ensure stabilization of RGSS localization at the upcoming new cell poles. We propose that RGSS hallmarks the site for assembly of a new elongosome. In the absence of binding of RGSS to MEC-processed peptidoglycan, anchoring of RGSS at the new cell pole is weakened. Therefore, RGSS might accumulate and promote formation of the new elongosome at the old cell pole. RGS proteins, including RGSS, are conserved in Rhizobialis. We therefore assume that positioning of the growth pole in S. meloti and other unipolarly growing members of Rhizobialis is determined in a similar manner. I will now switch to the application part and present some ongoing work of bacterial genome engineering, which takes advantage of our fundamental research of cell biology of alpha rhizobia. This also touches questions of spatial organization as one factor to take into account when engineering bacteria. One approach for adding additional genetic content to bacteria or to sort genes into functional groups is establishing synthetic secondary replicants, which in contrast to classical plasmids integrate tightly and stably into the bacterial cell cycle. Although most bacterial species carry a single chromosome, about 10% of the complete genomes in the databases contain more than one type of chromosome. The group of alpha proteobacteria rhizobia is a prominent example for multipartite genomes. Many alpha rhizobia have, in addition to a main chromosome, up to 6 additional secondary replicants. These are either classified as chromids or megaplasmids, depending on the presence or absence of essential genes. The Zynerizobium meloti genome is composed of the main chromosome, the chromid B, and the megaplasmid A. Due to partitioning systems and regulatory integration into the cell cycle, these secondary replicants are very stably maintained. However, with brute force, colleagues in Canada generated strains cured for one or both secondary replicants. In case of a chromid, a few core essential genes had to be translocated to the main chromosome prior to curing the chromid. In my lab, we engineer genome architectures to learn about organizational constraints and to use this knowledge to aid design of genome organization. This includes genome reduction, but also reorganization by rearranging replicons and by establishing additional secondary replicons to modularize genome organization. Most of the work that I will present on this topic was done by two very talented PhD students, Johannes Döhlemann and Marcel Wagner, and supported by a number of undergraduate students. In S. meloti, the chromosome has a longitudinal arrangement. The replication origin of a chromosome localizes at the old pole, and the origins of megaplasmid and chromid localize in the subpolar compartment of the old pole. We demonstrated by tracking the origins with fluorescent labels that duplication of a chromosomal and megaplasmid origins of replication is spatially and temporally uncoupled and occurs only once per cell cycle. The chromosome origin is segregated first, starting close to the old cell pole, followed by the A and the B origins, with segregation starting around mid-cell. We then asked what genetic elements are required or sufficient to confer this tight integration into the cell cycle and the faithful maintenance of the secondary replicons. This is mediated by the RepABC module. It encodes the replicator initiator RepC and the partitioning proteins RepA and RepB. The region further includes partitioning sites that are bound by PAB. The origin is in the RepC coding region. Expression of these genes is tightly negatively autoregulated at the transcriptional and post-transcriptional level. As a tool for changing the number of secondary replicons and as a vehicle to trigger genome rearrangements, we have generated a set of modular RepABC-based vectors. In addition to RepABC regions collected from members of the alpha proteobacterial order Rhizobialis, these vectors contain various standardized modules for selection, for replication in E. coli, for mobilization, and for insertion of cargo. For each alpha proteobacterial host, appropriate RepABC regions have to be identified, as they have to be compatible with the chromosome and if present with indigenous RepABC type replicons. For S. meloti, we have identified 5 RepABC regions that mediate stable maintenance, even in the absence of selective pressure. And we showed by quantitative PCR and fluorescent labeling that the replicons are single copy. So far, we have integrated 3 PABCs in parallel, in addition to the 2 indigenous RepABC secondary replicons stably into the S. meloti cell cycle. We are continuously extending the set of compatible RepABC parts for alpha proteobacteria in my lab. So recently to methylotrophic methylorubrum and photosynthetic Rhodobacter, which are interesting chassis for applications in biotechnology, and to Dinoroseobacter, which is a marine alpha proteobacterium. Knowing about native occurring replicon fusions in Rhizobia, we wanted to investigate effects of such genome rearrangements in more detail. Therefore, I would like to share some results from targeted fusions of the native replicons. This shows a design of fusing both secondary replicons and all free replicons while maintaining a ratio of replicors, a coding sequence, orientation, bias, so the CDS skew, and a distributional pattern of COPS motifs similar to the configuration in the wild type. COPS is the FTSK orienting polar sequence motif, which is involved in chromosome segregation by FTSK. The frequency of this motif usually increases the proximity to the terminus, which was maintained when designing these replicon fusion strains. enriched and defined minimal medium, the strains with stably fused replicants showed normal cell morphology and growth. We also demonstrated that all three origins of replication are active in the fusion strains. For time reasons, I'm not showing the data on this. In activating the origins of the secondary replicants individually or together in the fusion strains resulted in viable strains. Strains with bipartite genomic DNA configuration with only one origin to replicate chromid megaplasmid fusion grew slower than the wild type. And also strains with monopartite genomic DNA configuration lacking one or two of the origins grew slower than the wild type. Although growing slower than the wild type, cell morphology was unaltered. Therefore, we looked a little bit deeper into these fusion strains. We then analyzed the spatial organization of the monopartite genomic DNA in the triple replicant fusion strain compared to the wild-type configuration by mapping origin and terminus regions and a number of further genetic loci along the monopartite DNA. Here, I'm showing only snapshots of origin and terminus positioning in the replicant fusion strain. Further, we developed in collaboration with a group of Peter Lenz from the Theoretical Physics Department in Marburg, a model to predict spatial genome configurations. This is an example configuration of simulations consolidating physical principles like self-avoidance and compaction of DNA and spatial constraints for specific genome positions. The overall result of our analysis was that in the strain with monopartite triple origin configuration, origins of replication positioned as in the wild type. Also, the spatial and temporal dynamics of origin partitioning was not affected by this genome configuration. Here we see a small series of frames from a time lapse in the fusion strain with triple color labeled origin regions. The preferential pattern of chromosomal origin segregation, first followed by segregation of a megaplasmid origin and then of a chromid origin, was conserved in the triple replicant fusion strain, as was the spatial pattern of segregation when comparing wild type to the strains with monopartite configuration. In the last minutes of my presentation, I'd like to close my tour through our recent research by showing one application example from the field of DNA-based information storage. So it's just a little bit a snapshot into this application project. Why storing digital information in DNA? This is attractive for long-term archiving because of the high storage capacity that is a million times denser than in a classical flash memory. When properly stored and protected, DNA can be stable over thousands of years. Examples for this are DNA from fossil bones. And synthesis, copying, and reading technologies are well established and costs are constantly dropping. Using DNA as information storage medium was first proposed as early as in 1964 by Michael Neumann. But it took until 1988 until the first demonstration was published, at that time with an oligonucleotide with a massive length of 80 nucleotides. Since then, the field has developed with the developments in DNA synthesis and sequencing technologies. Recently, with pilot studies with more than 100,000 megabytes stored in DNA. The workflow usually includes coding of digital information in a DNA sequence, DNA synthesis. Most approaches explored so far are based on oligonucleotides that are stored in vitro. Only a few studies used in vivo systems, and all these were based on bacteria with only small pieces of information stored per cell. The information cycle is completed both for in vitro and in vivo systems by sequencing and then by decoding of the sequence to the original information. Either the whole library is sequenced or sub-libraries are targeted by sequencing. In the MOSLA research consortium with colleagues from the informatics, we are developing and evaluating codes for storage in DNA, both using in vitro and in vivo approaches. I shortly would like to give you some snapshot insights in our in vivo work. Theoretically, about 70 micrograms of lyophilized bacterial cells with either just 200 base pairs or 1 kilobase information coding DNA per cell would carry 100 gigabyte or 500 gigabyte of additional information. What I will present is work of Tolga, a PhD student from my lab in collaboration with Marius and Michael from the informatics groups of Dominik Haider and Bert Freisleben in Marburg. To test storage of larger pieces of information in vivo, Marius and Michael encoded the brother Grimm's fairy tale, The Sleeping Beauty, which you may know from a Disney movie, in a simple code and in two different fountain code-based codings. These two had computational error correction capacities implemented in the code. Tolga then realized the codes in DNA and implemented it in Zynerizobium maloti, either assembled to a 20 to 25 kilobase region of DNA or separated in small fragments. Formats were integration in the different S. maloti replicons or in a PABC vector. This resulted in a set of S. maloti strains, all carrying the fairy tale information in different biological and in different coding versions on the native replicons and on additional PABCs. In ongoing work, we are now testing long-term stability dependent on codes, biological formats, and various environmental conditions, and we are testing the performance of the implemented computational error correction methods. With that, I would like to wrap up. I showed how a quorum sensing system probably uses modulation of pulse frequencies of AHL synthase gene expression to integrate each cell's physiological state and need for behavioral adaptation in a collective decision-making process. I also reported that FTSN-like protein and amidase are required for determining the growth pole in Zynerizobium maloti. Further demonstrated engineering of the S. maloti genome architecture by targeted replicon fusions and by implementing PABC vectors, and an application of Zynerizobium maloti and artificial secondary replicons as digital information storage media. With that, I'm at the end of what I wish to present today. The presented work would, of course, not have been possible without all the dedicated co-workers in my lab and without the help of many collaboration partners, and of course, without generous funding by various funding institutions. Thanks for staying with me, and thanks for listening.
Well, thank you, Anke, for a very interesting and clear talk, as you always do. I'm sure that there are going to be a lot of questions over here. So if you want to do a question, please raise your hand and come over here to do it. Yes, please. Jesus Montiel.
Thank you for the talk. And I was just wondering if you ever test if there is any role of this RGS or amidase in bacterial differentiation during nodular organogenesis in Medicago?
No, we haven't tested it because all of these proteins are essential, or they have very severe growth phenotypes. So it would be rather difficult to manipulate the levels and activities of these proteins in the bacteroid. So the only way to do this would have a very tight system where we could switch between different versions in the bacteroids. But what you can see if you look into data from transcriptome or proteome studies, the transcriptome studies with microdissection from Toulouse, for example, very nice study. You can see that that the levels of these the levels of a transcript, levels of proteins are changing. And that fits very nicely also to work from Emanuele Biondi and from Peter Merget that indicate that the cell cycle is disturbed during bacteroid differentiation. And of course, also the cell growth, because that is tightly coordinated with that.
Okay, further questions? Come on.
Thank you very much, Anke, for the presentation. I didn't get really the last point with the data storage. Could you be, I don't know, I'm maybe too ignorant to get it, but how, is there a code? How you, well, how you transfer the digital storage to the nucleotide sequence?
Okay, yeah, there are several codes which are developed by our collaboration partners in the informatics, and there are also several codes published which we use and benchmark against our developments. Coding means that the binary information, so zeros and ones from the digital files are converted into a DNA sequence. And you can't do this in a simple way by just giving every block of a 0 and a 1 a block of a fixed DNA sequence, because you would end up with molecules first which may be difficult to synthesize or to amplify or to stably keep in a cell or in an in vitro system. So you have to avoid, while you encode this, that you get such structures or promoters or other elements which you do not wish to have in a cell. And in addition, like with every other storage medium, so magnetic or whatever it is, of course, during synthesizing, writing and reading, errors occur. And these errors need to be corrected. And in computer sciences, there are quite some sophisticated error codes already developed there with only a little bit of additional storage capacity, which you have to spend. You can correct errors in your storage medium to retrieve the full information back. These error codes have been developed, for example, for DVDs or for wireless transfer of information. So we are using it and depend on it every day. And what we are doing is we are further developing these codes and establish new error codes which are specifically designed for the needs and the problems which occur when you handle DNA. I could give a whole lecture on this if someone is interested, but that's how deep I can go in a short answer.
Yes, Osama?
Well, thank you. I had a question about the polarity experiment. Does the accumulation of those two substances, I think the RGCs,
louder?
Is it not working? It's working. In the new cell divisions after the first one, does the accumulation of those substantiation will be randomly in one of the pole growths, or is there a preference for the new growth pole?
No, there is a very, very strong preference for the newborn pole. And our idea how this is achieved is that parts of the RGS proteins, or a large part of the RGS proteins, is already at the septum when cell division occurs. And it stays at the newborn cell pole and sort of hallmark for recruiting other new pole characterizing proteins to the site. So it's sort of inherited. And of course, also dependent on the properties of the pole and our ideas also of the peptidoglycan structure at the newborn cell pole.
Okay, thank you.
Anyone else? Well, I have a question. Regarding this, your efforts about genome architecture, the fusion of the 3 replicons into a single replicon, does it matter the order of the different segments or the orientation?
Yes, definitely. So I have only showed you one example of a very stable design, which was a design where we tried to follow all the rules and all what is basically known from genome architecture and to keep orientations and directions and enrichments of motifs in a sort of multiply-like manner. But we also made a large number of different types of fusions where we made very asymmetric fusions, or also inverted the orientation. You also see an inversion already when we remove the PSMA and PSMB replication origin, if you make a Rep-C deletion in the triple replicon fusion strain, because what you end up with the mono origin DNA molecule, the cells are viable, they grow, they have some problems. I couldn't show this, and we think that to some extent we understand these problems. And here you have a largely imbalanced left and right replicor, and of course by removing the Rep-Cs, the orientation of the arms of replicors are now inverted with respect to the active origin.
Yes, I was curious of that because
not everything is possible. I think a few years ago there was a publication about a native fusion of the free Zynerizobium and Loti replicons by homologous recombination between S elements. And actually this, what happened just by chance, is the design which is most stable in our hands, because it keeps all these balances.
Yes. Yes, I'm familiar with that construction. Rafael Palacios, Georgina Hernandez participate in that. But we were also suspecting that alternative configurations probably were not available. There are several factors. One is replication, the balance between different replicors. Other factor can be the orientation of the major transcriptional units, and a well-known.
Yeah.
In a theory,
right, and we really see if we remove from this triple replicon fusion, if we remove the PSMA and PSMB origin, we see in a marker frequency sequencing, which we have done, that the progression of a replication fork is not as smooth along these than long inverted replicor arm.
Yes, thank you very much. Well, if there are no additional questions, we have to move on. Thank you, Anke.
Thank you.
And the next speaker is just me. You're stuck with me now. And if puedes ponerla. Can you hear me well? Yes. Okay.
Yes.
Perdón. ¿Qué pasas? Okay. What I'm going to do with you today is to talk about a sizable part of my academic life and the academic life of my coworkers at the program of genome engineering. It's difficult to concentrate that into a talk, and I christened that on plasmids and chromids, dynamic components of rhizobial genomes. I have to say before starting that I'm here at the center because when I was a student, a bachelor student, I went into the lab of my professor and tutor, Jaime Mora, and with the unofficial direction also of Guillermo Dávila and the collaboration of Rafael Palacios. The excitement of the research that was being doing it that way at that time, and also the motivation that they put into the research was impossible to resist. When I knew that there was going to be a new center at Cuernavaca, I said, count me as the first one. I arrived to the center 1 year later of the start of the center, but it has been a very pleasant experience ever since. Not only I have a depth of gratitude with them, also with the co-workers that have collaborated with me at the program of genome engineering. Susana Brom, Alejandro Garcia de los Santos, Lourdes Girard, just for a while, Jaime Martinez, have been part of the constant workers that have been with me during this trip. But also I have to say that most of the work that we have done has been in collaboration with the rest of the center. In a very quick count, we have collaborated with more than half of the researchers present in the center. We were attracted to come here because of the nitrogen fixation process. And as you know, well, this bacteria has a free life, it nodulates plants, and it makes a strong difference if the plant grows in the absence of nitrogen. If there is no nitrogen, the plant practically doesn't grow, but if you have nodulation and nitrogen fixation, you can see that the plant is green and healthy. That in itself should have been a good motivation, but it was not the only motivation. Almost from the start, we were attracted by the unusual characteristics of the rhizobial genomes. As Anke has told you, there are a lot of extrachromosomal elements. In the bacterium that we work with, Rhizobium 6 large plasmids that goes in size from 180 kb to 600 kb. There are rhizobial species that have very large replicons of 2,000 megabases or even more. A sizable fraction of the genome that can go from 30 to 50% is in the form of what it looks like plasmids. For someone trained in E. coli that has a very normal chromosome, a very tiny plasmids, to find this is quite intriguing. There is a structural variation of those plasmids even within a single species. This pattern, you will not find it into another member of the species. It's going to be a scrambled arrangement, a very variable arrangement. One of the plasmids, in this case, this one, it was interesting for those involved in nitrogen fixation. It's the symbiotic plasmid that contains most, but not all, the genes involved in modulation and nitrogen fixation. So the question was quite clear. What are the functions encoded in those plasmids? How to reach for clues of their function in the times when it was impossible to sequence the whole genome. That was one of the group of characteristics of these genomes, but the second one was the presence of extensive gene repetition. Trained in E. coli, you are trained to say most of the genes are single copy. Well, except for the ribosomal, but there is just a single species, exception. Even from the start, from the center, Rafael Palacios found repetition of the whole set of nitrogenous genes. The HDK genes are repeated twice into the symbiotic plasmid. Those are exact copies. The nucleotide sequence is identical between this sector and this sector. And there is a third copy located in the center that it's only comprised by the NIFH gene. The third copy, so far to the NIFH gene, is completely identical also to the other two. That occurs for the nitrogenous genes, but it's not the only place where it occurs. It also occurs in other parts of the genome, not of the nitrogenous genes. Other genes are also repeated. In a quick estimation, repetitions of genes or sequences can represent up to 5% of the genome. Some of the repetitions are identical, and you can explain that by saying that they were generated very recently in evolutionary times. That or that there is a very active mechanism that is keeping them identical. The questions in that regard, what are the roles of those reiterations and what consequences they may have for genome stability? Those have been the major questions that have guided our research over the years. Regarding plasmid-associated functions in rhizobium, Susanna Broad devoted a lot of work to that. And what she did was very direct. Let's try to eliminate each one of the plasmids. She did that first by random approaches. Take a lot of different isolates of the same strain, single colony isolates, and screen those for cells that have lost the plasmids. It can work only if the plasmids are unstable. And she succeeded to get some of the isolates, some loss of some of the plasmids. But she did also something additional. She introduced a system that allows for positive selection for loss. And then she succeeded in eliminate every one of these plasmids individually. The only plasmid that she was unable to eliminate was plasmid E, this one of 500 kb. And keep that one in mind. There is a plasmid that it was impossible to eliminate. For the rest, you have phenotypes that are consistent with what we were expecting. For the symbiotic plasmid, and you can see that with this color key, you have problems. If you lose the plasmid, you do not nodulate, you do not fix nitrogen, and you don't have conjugative transfer of the own plasmid. Nothing surprising. It's an accessory plasmid. The same goes with plasmid A, that the only function that it carries is conjugative transfer of itself. Those are the accessory elements of the genome. But for the rest of the plasmids, interesting things started to happen. For instance, loss of plasmid B, the smallest of the plasmid, causes a problem in LPS biosynthesis. Even the appearance of the colonies changes in the absence of that plasmid. In a quick summary, there are variable frequencies of plasmid loss, but in almost every case, frequencies of loss are above the spontaneous mutation rate. In some cases, loss is quite frequent, 10 to the -2 or something like that. Some of the plasmids, like plasmid A and plasmid D, are classical plasmids in the sense that only encode functions that extend the ecological niche of the bacteria. Others are more mysterious. Some plasmids encode functions that are relevant to central metabolism. For instance, in plasmid B, you have LPS genes that participate in formation of lipopolysaccharide. In the same plasmid, you have also genes that participate in biosynthesis of a vitamin, thiamine in particular. And that is work of the names of the people that work in that are put in here. The only catalase gene that is located in the genome is in plasmid F. Genes for biosynthesis of pantothenate are located in plasmid F in particular. When I said that genes for these functions are located in plasmids, I am not implying that all the functions, all the genes necessary for a particular function are located in that plasmid. A more common situation is part of the pathway is located in the plasmid, the rest of the pathway is located in the chromosome. So the plasmid and the chromosome have to cooperate in order to achieve a function. Susanna Brom did something additional that it was very interesting. She applied the technique to eliminate the different plasmids in a recursive manner. She eliminated first one plasmid, and then she proceeded to eliminate the rest of the plasmids. So she had derivatives that like 1, 2, 3, 4, 5, and that's all. She ended up with a derivative that only contains the chromosome and plasmid E, and a small version of plasmid E. What she found was very interesting. Loss of almost each plasmid individually, with the exception of plasmid B, didn't cause too much trouble in terms of work. But when you started to combine the different losses, the growth rate descended precipitously. As marked as the strain that only carries the chromosome and part of plasmid E has a duplication time of 8 hours compared to the 2 hours of the normal wild type strain. So these guys, these plasmids, are integral components of the bacterial genome, of the rhizobium genome. You cannot lose them impuniously. They are needed for the growth of the bacteria, are adapted to that bacteria. So that goes against the normal vision that people has of plasmids in different organisms. That is a kind of a carriage, as something that the plasmid has to pay for its residence. In this case, those are components that are adapted to the presence in the rhizobium genome. Regarding DNA repetition, what we have found over the years is that repetitions in genes, for instance, of the genes for nitrogen fixation, cooperate for function. If you have 2 copies and you take one of the copy, you have half of the function that you had before. If you mutate the other one, you have half of the amount of the function that you had before. So these 2 iterations can cooperate, and those are exact repetitions. But we have found also examples of, particularly with non-identical repetitions, when there is some functionalization or even neo-functionalization. The reiteration, the repetitions of a gene adopt new functions. And there are examples in the literature about that. There is an additional consequence of the presence of repetitions in a particular gene. Those become targets for homologous recombination. Recombination between these repetitions can generate all sorts of rearrangements in the genome, and this is exemplified by recombinations in the symbiotic plasmid. Recombination between these 2 repetitions can generate deletions of the symbiotic plasmid at a very high frequency. As you can see, at frequency of 10 to the minus 4, you lose all this region, generating a smaller plasmid. The problem is that that deletion derivative has lost all the genes for nodulation and nitrogen fixation. So the resulting strain is unable to do the normal biological function of a rhizobium. We have found that this generation of this particular deletion is a major cause of inoculant failure in industries that produce biofertilizers. There are several examples of this kind of deletions that have accumulated in the strains that the companies has. Not only that, another variation of recombination can generate now the multiplication of the symbiotic region. Frequency of 10 to the minus 3, you can end up with a large duplication of the whole symbiotic region, or a triplication, or even higher order amplifications. We have succeeded in growing the size of this plasmid from 3,390 kb up to 1.5 megabases in size, just by homologous recombination. Sometimes the structures that lead to those rearrangements are nested structures. I have referred only by recombination between the nitrogenase operons that are located in here, and I have referred to deletions or amplifications that affect this region. But Close to those, there are also other repetitions that overlap in extension with the first one. So you can have deletions or amplifications of this type or of any of those 4 types. That is common in the symbiotic plasmid and in other replicons. It's not a general rule in other parts of the genome. Now in synthesis, what we know is that repetitions are quite common in plasmid B and the symbiotic plasmid and in plasmid A. For the rest of the genome are infrequent occurrences. Apparently, the symbiotic plasmid and plasmid A accumulate most of the repeated sequences that are present in this genome. Most, but not all. This is possible also in other parts of the genome, including the chromosome. Just to summarize, variation in the structure of the symbiotic plasmid by recombination, you can get deletions at frequency of 10 to the minus 4, duplications at frequency of 10 to the minus 3, all that is above the frequency of mutation. You can get also reversion of those duplications with a very high frequency. Any recombination between these 2 duplicates can revert that to the original state. You can get inversions also by recombination between these 2 repetitions. This is an infrequent event, 10 to the minus 7. And perhaps this is related to the things that I was discussing with Anke just a few minutes ago. The orientation of the elements can be detrimental for function or stabilization. So that is also one of the evidences that you cannot move the organization of the genome as freely as we suspect. We can get also cointegration with different elements. Cointegrations between plasmid A and the symbiotic plasmid. This plasmid is interesting. It's a conjugative plasmid and conjugates as hell. So when you have cointegrates like this, the whole symbiotic plasmid becomes conjugative with a very high frequency. Or you can get cointegrations of just part of the symbiotic plasmid, the top part of the symbiotic plasmid, the symbiotic one, with the whole plasmid A, making a smaller and very nice symbiotic plasmid. I have devoted a lot of work to that, a lot of my life to understand the mechanism in that and the rules that govern this, perhaps with the same aim that Anke has. Try to construct a rhizobium with a minimized genome. Think of all this in that direction. Plasmid transfer by conjugation becomes also very interesting, and that has been part of the work of Susie Brown. P42A is a self-conjugative plasmid, and the frequency of transfer is very high, 10 to the minus 2. The system is controlled by quorum sensing in a manner similar to the one that Anke has described. The symbiotic plasmid has a conjugative system, but this system is silent. Normally, it doesn't work under free living conditions. For transfer of plasmid A, you can do it by yourself. But if you try to transfer the symbiotic plasmid, there is also the possibility of the cointegrate that I have described just a few minutes ago. Cointegration between these two plasmids, making now the symbiotic plasmid able to be transferred by conjugation. You can have resolution of this cointegrated structure that was generated by site-specific recombination by reversing the process that generates this, or you can recombine with other repeated elements, making now chimeras, plasmid that has parts of plasmid A and parts of the symbiotic plasmid. There is a lot of plasticity in the organization of these genomes due to these processes. Susie has characterized over the years 3 additional conjugation systems. Each one of them is controlled by quorum sensing. And she had found also instances in which conjugation requires cooperation between 2 different plasmids. In these systems, if transfer of a single plasmid occurs, it becomes a stable component in the gene. And there is at least one case, a possible case of what we call genogenic silencing, transfer of a novel element into a new genome and silencing of genes into the recipient gene. Susie then asked something interesting. We have seen so far conjugative transfer only in the free living state. But it's quite obvious that rhizobium can invade plants and establish a nodule that is full of bacteria. Is conjugation possible inside the nodules? The density of the cells is going to be very high. Those cells are going to be protected in a very high density. If it's possible, it will become a way of diversified genomes inside the nodules. So what Gonzalo Torres Tejerizo, a former postdoc in the lab, did was to label the chromosome with red fluorescent protein, plasmid A with the green fluorescent protein, and inoculate with this strain, plants of bean, inoculating at the same time a recipient strain. If conjugation occurs inside the nodules, the hallmark of that is going to be a cell that has only the green fluorescent marker, not the red fluorescent marker. When you transfer this into a recipient, you end up with only this. And that can be screened by microscopy. This is an example of the kind of thing that she has done. Transconjugates in this case are identified by the green fluorescence, not the yellow fluorescence that is the one that you can get when you have red and green. And this is on the root surface, but look inside the nodule. Most of the cells that you have inside the nodule are transconjugates that have conjugated the plasmid inside the nodule. We think that it was inside the nodule because what Susie and Luis Alfredo Bañuelos did was to try to suppress the transfer by putting an essential gene for conjugation, TRI, under the control of a promoter, the NIFH promoter, that only works inside the nodule. When she did that, no transconjugates were seen outside the nodule. Most of the transconjugates were seen inside the nodule. They did also the converse experiment. They put an inhibitor of conjugation under the control of the NIFH promoter. Now the inhibitor is going to be expressed only inside the nodule, suppressing conjugation. When they did that, no transconjugates were seen inside the nodule, but there were transconjugates outside the nodule, in the root or in the middle. So I think that this is very neat and demonstrates that conjugation is also an ideal niche to have diversification of the genomes of these bacteria. We have not abandoned the exploring of different functions in the genomes. We have also, in the case of Alejandro Garcia, now switching gears to try to understand other functions. The metal stress is the one that Alejandro chose with his group, and she has done a lot of work identifying the different genes and components that are important for tolerance to different metals. Metals are important in different soils, and their tolerance are relevant for the survival of the bacteria. We have a depth of curiosity. If you remember well, there is a plasmid that we were unable to eliminate. A plasmid that looked like essential, plasmid E, P42E. What is in there? We were expecting that in the moment that we had the full sequence, we will be able to identify the exact reason about why this plasmid was unable to be eliminated. When we got the sequence, it was interesting. But what we identified just by looking at the annotation is that a sizable fraction of the genes that are located in this plasmid participate in primary metabolism. The problem is that most of these genes, or the functions encoded by these genes, can be eliminated and the bacteria can survive in a rich medium. So why we were unable to eliminate that? Well, there is a gene that participates in cell topology, the MinCDE, but we did a mutation in particular of that gene, and the only thing that we got were small cells. But the cells were able to divide, and the derivative was quite happy to live. What we did then was to remove a large part of this replicon by using site-specific recombination, trying to delimit the region that was essential for survival. We removed most of the plasmid except for a small sector that was located here. The genes that are important for survival are only two. gene 1 and gene 24. All the rest can be eliminated and the bacteria lives quite happily, with some absences, but nothing too essential. But these two genes are apparently the main reason about why we were unable to eliminate that. Annotation wasn't very helpful to understand function. Regarding the gene 1, it's a hypothetical protein that is conserved in rhizobiales and has a helix turn helix, perhaps a regulatory protein, but there is no clue about function of that. The other is part of a two-component system in which we have the sensor element, but we do not have the response element. So we have half of the system, the response element has to be located somewhere else. Recent data in our lab have revealed that this gene has an interesting function, and we have rechristened this gene as RDSA. It's a gene that participates in cell division and shape. And let me show you that very quick. When you do not knockout mutations of gene 24, knockdown mutations, a mutation that reduces the amount of the transcript of the corresponding gene, you have, instead of the normal structure of bacterial cells, rod-shaped, you have spherical cells with a very high quantity. As high as 60% of the cells of the culture that has reduced expression of RDSA becomes spherical. When you complement the resulting strain with a wild-type copy of the gene, cells become now of the normal rod-shaped form. So apparently the gene controls shape of the cells. Not only that, it also controls the polarity of division that Anke told you about. Cells, a sizable fraction of the cells start to divide not through the new pole, but through the old pole. So there are problems of shape and also control of cell division in those bacteria. What we did was to do an RNA-seq analysis of the knockdown strain, the one that is affected in the level of expression of the gene. A large number of genes are affected, almost half of the, almost 600 genes. Half of those doesn't have a function, a recognized function. But for the rest, it was quite interesting. As you can see in this heat map, the ones that are located in blue or purplish, what they reveal is that there is reduced expression of those genes. The ones that are located in red has overexpression of those particular genes. And these were arranged in the form of function. All this block corresponds to cell division. This block corresponds to replication and nucleotide metabolism, peptidoglycan formation, all this, etc. So what we have is reduced expression of genes that participate in cell division and peptidoglycan formation. Both functions are essential for cell division and coordination of the poles. So there are also additional sectors that were affected in expression. For instance, all this corresponds to respiration of the cells. The cells have affectations in the levels of expression of the different complexes, of different respiratory complexes, including ATP synthase. So the message in general about this is that these genes participate in expression of essential genes for cell division and cell wall biogenesis, but also in coordination with other major functions as respiration. It's a global regulator that participates in coordination of the genes. We don't know at this time what part of the control is direct and what part of the control is indirect. We need to have the response element in order to know what parts of this control are due to direct control. And we are actively looking for the response element in this system. This was quite fast, but it was a sizable part of my academic life. And I tend to remember that we shall not cease from exploration. We started exploring something. And the end of all our exploring will be to arrive where we started and know the place for the first time. We don't know the whole place, but we are advancing in that direction. And thank you to T.S. Eliot for that. What will be in the future? Their own research is defining what we are going to do. And I'm using the phrases of Theodore Rothke by saying, I wake to sleep and take my waking slow. And I learn by going where I have to go. Thank you. And I think that I overextend a little. Is that no? Okay. Well, we have time for 1 or 2 questions if you want to do that. Okay, if not, let me introduce Pablo Vinuesa, my coworker, and say something. Pablo studied the PhD at Marburg University, the same place where Anke Becker is located. She did his PhD with Dietrich Werner, and she was young and smart. We met him for the first time in a meeting in Paris, of all places, about nitrogen fixation. And it was as exhilarating as it is always. We talk a lot about science, about that, and we have continued to do that ever since. He arrived to the center in 2001, just the day after the...
5 days after.
5 days? 5 days after the...
September 11th.
September 11th. Well,
that means I arrived on September 15th, which is also quite an emblematic date for Mexican.
But it has been quite delightful to have him here. We have collaborated with him a lot. But now Pablo is going to talk about something new in his research. That is called, well, you remove that. Accidental evolution. Pardon. The title of his talk is going to be Accidental Evolution of Opportunistic Pathogens in Natural Habitats. Stenotrophomonas as a model. Please, Pablo.
I thank you very much, David, for the introduction. I will continue in English as you started introducing me in that language. So let me use the opportunity to practice a little bit speaking English, which is not so frequent for me. Well, as David told you, I arrived in 2001, so it's now 21 years since I arrived, and I'm very happy to have, well, not only arrived at the center, but I have my family here, and well, I'm most pleased to be working here at the Center for Nitrogen Fixation. That was the center when I arrived, and now it's the Center for Genome Sciences. Did I hit some of the buttons? ¿Me lo pueden regresar? And well, I'm supposed to talk a little bit on the part of understanding genomes in this session. And we have been, in more or less recent years, working on a new group of bacteria that has never been worked on in this center. It's the genus Stenotrophomonas. And I will briefly explain you what we have learned and how we got to work with this bug first of all. All the adventure started with Luz. She came to our lab to start a PhD. She had already done a master's thesis at the center, at the Institute for Salud Pública, and so she had some experience working with antibiotics and so on. And well, taking advantage of her experience, we decided, let's try to understand a little bit more of the environmental dimension of the antibiotic resistance problem, a topic that was addressed briefly yesterday in the evening by, I don't see him. Yeah, sure. She's there. Okay, well, and as many of you, I'm sure you already know, antibiotics were introduced in the 30s, basically, and in the following decades, several classes of additional antibiotics were produced and introduced into the clinics. But all of a sudden, after the 70s, we had a gap. No more or new classes of antibiotics were produced, but instead resistances to all the previous classes appeared. So the first papers on the topic were a new resistant strain of Escherichia coli, whatever appeared, etc., etc. After the 2000s, basically all papers are multi-drug resistant or extreme drug resistant bugs, or even, as we heard yesterday, pan-resistant bugs like, for example, Acinetobacter baumannii and others. So we are really now in a perfect storm, as this special issue in Nature explains us, because the different lineages of multi-drug resistant bugs are just rising like crazy in the clinics, but the antibiotic production pipeline went just down. It's more financially more interesting for the companies to produce other sort of medicines, right, than antibiotics, which is a very complex topic and not so profitable. So we thought that studying the environmental dimension of this problem of antibiotic resistance was really a key to understand why there are so many new resistant strains arising constantly. And the basic model, the basic idea put forward by other colleagues and by ourselves as well, you can read here as a simple review in Spanish about it that I wrote some years ago. The point is that humans are putting an extreme amount of antibiotics, dumping them into the environment. And so we are selecting very strongly for mobile genetic elements that carry resistant genes. And these are spreading now through the landscapes by for example, irrigation with contaminated waters. Here we see in center Morelos, sugarcane fields irrigated with contaminated waters. We have here around 1 million thermotolerant E. coli cells in 1 liter of water. Imagine how many of those are in this, just in one field. So this is a huge planetary experiment. To give you some more figures, only in the US, over 18,000 metric tons of antibiotics are sold each year, most of which go not into the clinics. This goes into the animal production. And all the, all the residues from that is dumped into the fields or even into the waters, of course. And this is basically now mixing the microbiomes of humans and the animals we have, or we keep, with the microbiomes of native bacteria, many of which are from the same genera as these very critical opportunistic pathogens that we have in the clinics. So with Luz, we started sampling rivers around in Morelos, very heavily contaminated ones and also cleaner ones, as you can see here. And Luz, since she was an expert in determining antibiograms, she used many different sorts of antibiotics, 15 of 5 different classes, and also these disk assays to determine the antibiotic resistance profiles for each of the strains she isolated. And she isolated really lots. And I'm giving you here a snapshot of one of the samplings. This one was at Temisco, south of Cuernavaca. This is in the Apatlaco River, which is highly polluted. And what we see here basically is a heat map, is a clustering of the resistance profiles. So you basically put in a matrix, zeros and ones, depending if the strain is sensitive or resistant to that particular antibiotic. So you have a matrix and you can calculate distances out of that, and then you cluster it simply. No? And we have several metadata associated with the profiles, and you see here when you have, like in the diagonal, dark blue line, the distance is 0, it's the same profile, okay? Well, so what appeared here, we distinguished like 4 big clusters numbered here. The largest one here in the center contains strains with rather similar antibiotic resistance profiles, but look here, this is the number of resistances that Luz detected, and we had extremely drug-resistant bacteria here, some even pan-resistant. Both of them are Acinetobacter baumannii, our environmental ones, and the others are all Stenotrophomonas. So that is how we got interested in this genus, because it was extremely prevalent in polluted sediments. But we found Stenotrophomonas also in many other environments, also in clean ones, okay? Well, so what do we know about Stenotrophomonas? They are gamma protobacteria. They are close relatives to Xanthomonas. They are in the same family, Xanthomonadaceae. There are about 16 species described. but only one is really well known, and that is Stenotrophomonas maltophilia. Why is it well known? It is one of the globally emerging opportunistic multi-drug resistant pathogens that is causing severe problems in the clinics. It's now in the top 10 list of the World Health Organization of most prevalent opportunistic pathogens in the nosocomial environment. And well, it infects, like all these bugs, mainly immunocompromised patients, very particularly those suffering from cystic fibrosis, but as Acinetobacter and Pseudomonas aeruginosa, they can cause pneumonias and bacteremias, facilitated mainly by mechanical respirators and indwelling devices. No? That is a typical situation. Yeah? So they have also a very strong tendency to form tenacious biofilms, and well, they cause other sorts of illnesses, some also from soft tissues, etc. So it's really not a specialized pathogen. No? It can colonize very different niches, including, that is a very recent work that is appearing in the literature. They are able, at least some of the strains of Stenotrophomonas, they're able to survive the digestion by environmental amoeba. These are some of the natural predators. We think that this may have served as a selective force for different kinds of bacteria to learn how to survive within eukaryotic cells. And as a matter of fact, Stenotrophomonas maltophilia can invade epithelial cells. These are electron microscope images from human lung epithelial cells colonized, invaded by Stenotrophomonas. It can replicate, or seemingly always within vesicles, but recent evidence has also, in the literature, also shows that they can colonize macrophages and divide within them. So this is an organism with a broad ecological balance. It can live a free living life, but also associate with eukaryotic cells. So what do we know about genomes of Stenotrophomonas? I'm gonna now switch to this second part of the talk, and I will present you first of all, the genomic resources we have created. We have sequenced, right now we have 62 complete genomes. Of them, 36 are completely closed. These are figures similar to those of closed genomes you find in RefSec. Well, that was last year, actually. It's a little bit dated, the slide. And the tree shows the position of our isolates. It's in the outer circle. You see the position of our genomes, which basically span the known diversity that we currently know of the genome. So this is an important genomic resource. We have a huge diversity of these bacteria all around the tree of the genus, so multiple species, and at the same time, we have the organisms to work with. So we have also developed different tools to analyze genomes. A quite popular package is GetHomologs, developed with my colleague and friend Bruno Contreras, who was a postdoc here, a postdoctoral researcher in Julio Collado's lab some years ago. And well, the pipeline basically is able to perform basically all the computations you need in parallel and on a high performance computing environments to compute clusters of orthologous or paralogous groups of sequences, okay? And with additional, so this is what the main script does, but with other auxiliary scripts, you can analyze basically the clusters of homologous genes to define, for example, core genomes, pangenomes, and do different sorts of statistical analysis on them. And very importantly for our work is you can use some of these auxiliary scripts of the pipeline, of the package to identify, for example, lineage-specific gene expansions or lineage-specific gene families that are only found in one cluster and not in another of the group of organisms you are analyzing, okay? Some years later, we developed another pipeline, which connects very neatly with GetHomologs. This is GetPhylomarkers, which selects basically high quality molecular markers from the genome sequences, from the core genomes, to use them for phylogenetics, okay? It selects basically alignments that do not have signals of recombination, but have a good signal of phylogenetic signal. Let's reduce it like that. And we have used this pipeline, for example, to review the whole molecular systematics of the genus Stenotrophomonas, noting that there's lots of problems. All these black arrows here denote misclassified strains. Many strains in the databases are called Stenotrophomonas maltophilia, even though they are really not that. No, maltophilia would only be really this group down here. And you can also do different sorts of pangenome analysis and pangenome phylogenies, even under maximum likelihood or under distances. And we use all these tools very frequently to localize basically lineage-specific genes. This is a very important topic for what comes now. And all this software is open source. You can freely use it and download it from GitHub. So let's now see a little bit how we are using these tools and our genomes to understand how complex phenotypes have evolved in this group. And we are focusing mainly on antibiotic resistance and virulence. So this is a maximum likelihood phylogeny of the core genome for complete genomes of Stenotrophomonas, ranging, this is the entire known diversity of the genus. And well, here we have the outgroup. This is what we call the basal lineage. These are Stenotrophomonas that have smaller genomes and that have never been described as pathogenics. Okay, and here we have a second large clade, and you see here this arrow points to the group of strains that we call Stenotrophomonas maltophilia in the broad sense. And here you have these red squares denoting which of those are clinical isolates. And clinical isolates are only found in this clade. So something happened in this branch during evolution that made these organisms more virulent or capable of causing infections in humans. A first evolutionary trend, genome evolution trend, which is very clear, is that if you now sort the genome sizes from strains of the basal clades to until maltophilia, you see a very large increase in genome size. The smallest genomes here are only like 3 megas, and the largest are about 5 megabases. So there's a lot of DNA in difference. What happens here? The first thing we thought, oh, there must be lots of plasmids. Oh, I mean, I studied rhizobium, so I thought everyone has plasmids, or so if not, it's not interesting. And what first we thought a delusion, because not a single plasmid is in these genomes. But there are lots of mobile genetic elements in form of different sorts of islands. We have conjugative islands like ICE elements, mobilizable islands like these IMES or IMES, and when some integrons, etc. Okay, so what about multi-drug resistance? We use this reference phylogeny to understand now the evolution of this complex phenotype, antibiotic resistance. And in one of those papers, we learned that Stenotrophomonas maltophilia, the isolates in this clade, and only those were really the multi-drug resistant bugs. This is a multiple correspondence analysis, and you see in red, and perhaps you can see it, and this is the resistant phenotype for these 15 different antibiotics that was tested, and they all cluster within maltophilia. The other isolates from these other more basal lineages, they are much less resistant. And as a matter of fact, isolates from clade 1 are extremely sensitive. Here, for example, in this assay, they are sensitive to all the antibiotics, to all the beta-lactams that she tested. Another very interesting finding of Luz was that she discovered that only maltophilia strains had or expressed metallo-beta-lactamases. And these are the most critical enzymes in the clinical settings because strains expressing these enzymes are resistant basically to all sorts of beta-lactams, with the exception of aztreonam, and generally it's associated with many other resistant genes. So this explained for the first time, Luz demonstrated that it's only maltophilia in the sense as we defined it phylogenetically that are the strains that are really dangerous. And so we wanted to learn a little bit more about the molecular basis of this complex phenotype. And it's a complex phenotype because you can be resistant to a certain antibiotic by very different means. You have the classical enzymes that either hydrolyze or modify drugs, inactivating them. Okay, or the targets can be modified by other enzymes, or the targets can be mutagenized, but mutated and suffer spontaneous mutations and are then insensitive to the drugs. But very frequently overlooked aspect, for example, in Santiago's talk, not a single mention was done of this, is that there are lots of efflux pumps. And there are many different, well, 6 different families. There are membrane proteins that pump different substrates. But there's one class of them, they are called tripartite efflux pumps, which have the pump inserted in the membrane, but they connect directly to the outer membrane through accessory proteins, let's call them like that. And these are highly promiscuous machines to pump out all sorts of not only antibiotics, but many other chemicals, okay? So they have basically to take out the rubbish that these other pumps send to the periplasm and pump it out of the cell. So that's why they are so promiscuous. And this is a problem because if you have many of these R&D, efflux pumps generally makes the strain extremely drug resistant, and in a very general manner. It's not specific for just one class of antibiotics. That's the point. So I constructed hidden Markov models for all the different kinds of efflux pumps known, and run this through the genomes and to find what the elements, which of those pumps they have, and mapping all this information on our reference phylogeny revealed a very fascinating pattern. The basal lineages have very few pumps. These are pumps that are part of the core genome of Stenotrophomonas, but as the genus diversified, the new lineages, the new species emerging in time, were gaining basically new types of efflux pumps. There are some secondary losses, but the point is that if you count the number of proteins involved in efflux, Stenotrophomonas maltophilia has by large and very significantly the highest number of efflux pumps, explaining largely the high resistance displayed by this species. Regarding standard resistance genes, there are only few generally in a genome, as you can see here on the scale. In maltophilia, there are around, as a median, like 2 genes, always this metallo-beta-lactamase, and well, perhaps a few others. So that means that most of the resistance in Stenotrophomonas is intrinsic. It's stably encoded in the chromosomes in the form of these efflux pumps. But Stenotrophomonas strains can also acquire resistance via different mobile genetic elements, like, for example, integrons. It's in a low frequency, less than 3% of our isolates carry integrons, and only one is a one-class integron. But this is a very interesting one because it carried 5 different antibiotic-resistant genes, including this one here that encodes for an extended spectrum beta-lactamase. And this is from Stenotrophomonas acidaminifila, one of the basal strains, which I explained to you are highly sensitive to antibiotics. But this strain had a very strange resistance profile, and that's because it received this integron with all these different genes, which, when looking at the databases, are mostly similar to either Klebsiella or Pseudomonas aeruginosa genes, so clearly horizontally transferred. So we have developed further tools to analyze other mobile elements like the ICE and IME components. I'm not going to get into many details of that, but here just some summary figures. There are a lot for 409 genomes of Stenotrophomonas and Xanthomonas. These are complete closed genomes. We find a huge number, 480 of those elements. And in the inner circle, you see the IMEs. These are elements that can be mobilized, but in trans, while the eyes have all the conjugative machinery to move by themselves, okay? But they have a rather patchy distribution, and the pipeline is able then to clusterize these mobile elements that it finds and search in databases for close relatives, and so we can get a better idea of what are the types of ICE elements, what they encode, etc. And some of them are encoding, as you can see here, antibiotic resistant genes or metal resistances as well. The pipeline is able to perform also automatic comparative genomics. It's very, very efficient in time. And we see that these ICE elements have an open pangenome, meaning that they are extremely variable. You see highly conserved regions. These are the regions involved in integration, excision, replication, and conjugation, and all the other is accessory genes. So these are hotspots of genetic diversity within these mobile elements. And finally, this is the last topic. I'm just gonna go very quickly over it because this is evolution of virulence. And this is basically work of the students in my group right now, or are all PhD students mainly. Well, Julio and I don't know if he's around. Perhaps you can raise your hand. Yeah, there you are. And Stephanie, and of course with Javier, our technician, and Diana Vasquez as well. And well, there are many different sorts of islands, not only IMES and ICE, but also what we have now started as defining as virulence genomic islands. And here's one of them. Very tightly packed. Here we have an integrase. There's a type 2 secretion system, which actually secretes the only few known virulence proteins of Stenotrophomonas. These are proteases that are secreted in high amounts by the clinical strains. And here is a locus encoding a sort of a fimbrial, which allows the strains to attach to surfaces and form biofilms, and several autotransporter loci, all packed tightly together in this region, which we can exactly map where it inserted because it's not present in the basal lineages like Stenotrophomonas acidaminiphylla. So again, we constructed hidden Markov's models for all the classical standard secretion systems and surface appendages described for gram-negative bacteria and search for them in the genomes. And we find again the same pattern of accumulation of more and more of these secretion systems, fimbriae, etc., as the genus diversified, being maximal, the representation of these secretion systems in Stenotrophomonas maltophilia. So, as a phylogenomic prediction, we thought that this may explain, or this would predict that Stenotrophomonas maltophilia should be the most virulent strain, or the most virulent species in the context of the whole genus. And the last slide, oops. Yeah, ya está. The last slide actually proved this hypothesis. And this is work done mainly by Claudia Torres, with help also from Julio and Javi. They injected the same amount, about 10 million CPUs of different strains of each of these species across the diversity of the genus, and injected 10 microliters of it into larvae of Galleria melonella. And this is an alternative host to assay virulence of bacteria. And what they found, these are Kaplan-Meier survival curves that model basically the probability of survival as a function of time. And for the same inoculum dose, all the basal species like Acidaminiphylla, Umi, and Terra hardly kill the bacteria, the larvae, sorry. Whereas the Maltophilia down here, after 60 hours, all strains killed all of the bacteria, all of the larvae. So demonstrating that they are actually the most virulent strains. And so now Julio and Stephanie and others are doing the genetic analysis of selected loci, and on that we will talk in another occasion. And it would be all for my presentation. Let me just sum up. So we have a collection of environmental Stenotrophomonas strains isolated here in central Mexico. And we have generated 62 genomes of them of very high quality, and they span the known diversity of the genus. So this is an excellent model to study the evolution of complex traits at a genus level, not within a species, but in the context of a whole genus. Plasmids are rare in Stenotrophomonas genomes, but there are many different sorts of mobile genetic elements encoded in the chromosomes. which we are starting to study in a little bit more detail. And finally, our phylogenomic analysis revealed that efflux pumps and different or diverse secretion systems, fimbria, etc., have been accumulating along the diversification history of the genus, being maximum in Stenotrophomonas maltophilia, largely explaining, first of all, the high intrinsic resistance to antibiotics displayed by this species, and we think also this explains the much higher virulence of maltophilia on Galleria melonella. And we think that since all this happens out there in the environment, this finally is the way how, in a fortuitous way or accidental way, out there in the environment, bugs can evolve that can cause trouble for humans in the clinics. So the key questions here are really, for example, the efflux pumps, what are the real physiologically relevant substrates of them? In the clinics, we know, yes, they pump out antibiotics, but out there in nature, what's selected for their presence there? These are really tough and difficult questions that we eventually would like to address in the future. And the same happens for some of the virulence loci, which we think are really more involved in survival, for example, against predation, perhaps by amoeba or so. And well, that would be all from my side. Thank you very much for your attention.
Okay. Questions for Pablo? Come on, guys. Pablo, don't move. Can you elaborate about the idea about genes for virulence for survival in amoeba? What's the idea behind that?
Yes, I was very tempted to talk a little bit about that, but this is really work in progress, and but we have quite some nice progress already on that. There are a few papers appearing recently in the last 5 years in a very scattered way of the presence of Stenotrophomonas within different genera of amoeba, of free-living amoeba. Acanthamoeba is a very well-known model, but Vermamoeba as well. And so here, these are electron microscope images showing what seem to be replicating Stenotrophomonas within phagocytic vesicles that obviously never maturate into phagolysosomes, because if that would be the case, they would get destroyed. How they manage to manipulate vesicular trafficking, etc., within the amoeba, we don't know. We have already transfection systems set up for Acanthamoeba. We have first transfectants with expressing RAB7, which is a key protein in the vesicular trafficking along the phagocytic pathway, and very preliminary data, microscopic data, suggests that the Stenotrophomonas, at least our strains, a part of the population at least, does not get into these vesicles target or marked with RAB7, which would be an indicator of a late phagosome. No? And so how they escape to that, we don't know. But how they manage to do that is probably a key for this bacteria to eventually cause infections in humans, and as I showed you, for example, invade epithelial cells. How they do that? There's no data in the literature. That is why we got interested in Stenotrophomonas, because from the molecular side or genetic side, there's really hardly any knowledge about the virulence mechanisms. And particularly if you compare it with all the knowledge known for the other top 10 opportunistic pathogens that are listed by the World Health Organization. So we think we have a good model to advance our understanding in this line. Thank you for the question. Esperanza.
Hi, Pablo. In some cases, you find that pathogens lose some genes that interfere with pathogenicity. Have you also observed that?
No, we haven't. There's a very complex. I have only shown what looks at the beginning the most obvious thing, gene gains. But you're completely right. In one of the slides, I showed some secondary losses of some of the efflux pumps If that can have a role in variants, I don't think so. But we have not identified at this point a gene that could be a good candidate for saying, okay, after we lose it, we are more virulent or are able to colonize a different niche. We haven't looked at that yet. Thank you.
Anyone else? Just a quick comment. It's really scary if these guys found a reservoir to survive inside the amoeba. And in that same vein, perhaps it's too early to know, but the conditions in which Stenotrophomonas cause disease are the exact conditions that we're experimenting today. Immunocompromised patients with respirators and all that. Has anyone checked the distribution of Stenotrophomonas in patients, perhaps not in the ones that came out from the hospital, but the ones that has long COVID? Some of the symptoms are pneumonia.
I have not read a single paper about Stenotrophomonas in COVID patients, but it's obvious. With all the respirators that have been applied in these last 2 years, many polymicrobial infections will have taken place, and it's very frequent to co-isolate Pseudomonas aeruginosa with obviously Acinetobacter baumannii, but also Stenotrophomonas. And that's the point is you make a big avenue for the bacteria to enter because they are so resistant to disinfectants, etc. It's very hard to get them away, and they form these very tenacious biofilms, so you can hardly remove them. And if the patient is also under antimicrobial treatment, so antibiotic treatment, you eliminate a good part of the microbiome, and then these bacteria, even though they are not highly virulent, have just the opportunity. They clear the way for them, and they use the resources and make these terrible infections.
Okay, thank you, Pablo.
Thank you for your attention.
Thank you all the speakers for a set of nice talks, and all of you for your participation and attention. And I remind you, be back at 4 o'clock, either virtually or presentially, okay?
Lo entendieron todo, ¿no?
Bueno, antes de que se vayan, nos entregaron 4 medallas más, entonces aprovechamos de una vez de entregarlos, por favor. ¿Con quién empezamos? María Los Ángeles Moreno Ocampo, por favor, andas por aquí. Los grandes certificados que van a María Gabriela Guerrero Ruiz. Felicidades. ¿Quieres una foto? ¿No? Pero conseguí que lo pongas. Muchas felicidades. Y tenemos María del Carmen Vargas Lagunas.
Wow.
Felicidades. Y María de Lourdes Giracuesi. Que no se lo esperaba, creo.
Sí, es mía.
Muchas felicidades. Y bueno, queda una medalla más, pero la persona no se encuentra, mañana la vamos a entregar. Sí, pues muchas, y pues